Dear students, welcome to all of you. Today we are here to discuss biochemical coevolution. First, the introduction. Biochemical coevolution may be defined as an evolutionary change in a trait of the individuals in one population in response to a trait of the individuals of a second population, followed by an evolutionary response by the second population to the change in the first. Biochemical coevolution is a kind of cumulative evolution in which the exchange of genetic information among the kinds is minimal or absent. Biochemical coevolution is the joint evolution of two or more non-interbreeding species that have a close ecological relationship, such as plants and herbivores, large organisms and their microorganism symbionts, or parasites and their hosts. The evolution of one species in the relationship depends in part on the evolution of the other through reciprocal selective pressures. Plants have evolved many mechanisms to defend themselves from herbivores. The most obvious are morphological defenses, for example, thorns, spines, and prickles, which play an important role in discouraging browsers and plant hairs, especially those that have a glandular, sticky tip, deter insect herbivores. Some plants, such as sedges, deposit silica in their leaves, both strengthening and protecting themselves. Significant as these morphological adaptations are, the chemical defenses that occur so widely in plants are even more crucial. Best known and perhaps most important in the defenses of plants against herbivores are secondary chemical compounds. Secondary chemical compounds are biochemicals produced by plants in response to selection pressures. These pressures may be from herbivory, competition, or the need for pollination. As plants produce compounds to exchange their survival, predators, competitors, and pollinators react and evolve means of adjusting to these compounds. Secondary metabolites are often under high selection pressure, causing individual compounds to have very limited distributions and making them useful in determining the evolutionary relationships between taxonomic groups. Presence of secondary compounds influences the activities of organisms interacting with the plants and over long periods of time the influence evolution of these species. Secondary compounds, once thought of as waste products, include an array of chemically unrelated compounds such as alkaloids, for example, morphine, cocaine, caffeine, nicotine, terpenoids, for example, essential oils, taxol, rubber, cardiac glycosides, isoprene, phenolics, for example, flavonoids, tannins, lignins, catechols, salicylic acid, quinones, for example, coenzyme Q, and even raphides, for example, needle-like crystals of calcium oxalate. The presence of some of these compounds can even characterize whole family or group of families of flowering plants. In nature, these chemicals play a major role in restricting the palatability of the plants in which they occur, or even forcing animals to avoid the plants altogether. Being toxic, growth inhibitory, repellent, or deterrent, these compounds can reduce fecundity, slow development, reduce digestibility, or have synergistic effects. Pollination and fruit dispersal systems have developed particular coevolutionary patterns in which many of the possible variants have evolved several times within a particular plant family or even genus. The resulting array of forms gives the plants a wide variety of pollination and fruit dispersal mechanisms. In the case of biochemical relationships, however, the evolutionary steps have been large and definitive, and the whole families of plants can be characterized biochemically and associated with major groups of plant-eating insects. These biochemical relationships appear to have played a key role in the success of the angiosperms, which have a vastly more diverse array of secondary plant products than any other group of organisms. The idea of reciprocal stepwise coevolution proposed by Ehrlich and Raven in 1964 involves a continuous evolutionary arms race between plants evolving new secondary metabolites and herbivores evolving adaptations to these compounds. Later on, attempts to explain these observations eventually resulted in the theory of phytochemical coevolution. The theory states that the plants are selected to produce secondary chemicals in response to herbivore feeding, whereas herbivores respond by evolving disarming mechanisms. A byproduct of this disarmament is the loss of ability to feed on other plant species. With the passage of time, plants thus become more toxic and herbivores become more specialized. Plants that develop novel defenses enter a new adaptive zone and undergo phylogenetic diversification, and at the same happens to herbivores that develop novel disarming mechanisms. This so-called escape and radiation may be largely responsible for driving the evolutionary process. As the plant species possess a novel chemical diversity, the selection for disarming mechanisms increases. Similarly, diversification of herbivores with disarming mechanisms increases selection for new or stronger defense chemicals. Now, the anti-herbivore mechanisms. It has been suggested that the enormous diversity of phytochemicals within the plant kingdom makes it impossible for one and the same insect species to exploit all plant species. The number and diversity of host plants utilized by different phytophagous insects are highly variable, and the selective pressures acting on them are still poorly understood. Many hypotheses have been proposed to explain variations in host specificity among phytophagous insects. For example, there are those based on plant traits like plant defenses, plant appearancy, plant abundance, plant nutritional value, those based on insect traits like degree of intimacy with the host plant, neuronal capacity of the insect, and finally those based on natural enemies traits, for example, the impact of generalist predators. Anti-herbivore chemicals may have a wide range of effects on herbivores. Many compounds merely deter grazing. Crystals produced from calcium oxalate may be ejected from the vacuoles of cells along with proteinaceous toxins, causing tissue swelling in the mouth of an offending herbivore. Many monocotyledonous plant families such as Liliaceae, Heliconiaceae, and Ericaceae produce this type of anti-foraging device. This type of defense is especially notable in young tissues that have not developed the toughness found in mature leaves as a herbivore deterrent. The tannins produced by red oaks in response to gypsy moth attacks, reducing further herbivory. Continued feeding on plants containing tannins would lead to slow starvation of a herbivore, as its digestive systems could not absorb proteins. Beavers provide another example of the interaction between plants and animal evolution. Some of the beaver's preferred foods include species that are unpalatable or toxic to other mammals such as bracken fern, nettles, thistles, and skunk cabbage. This gives the beaver a largely uncontested food source that may involve a metabolic cost to the animal. Other anti-herbivore chemicals result in effects more severe than more deterrence of feeding. Alkaloids such as caffeine, nicotine, and strychnine are potent anti-herbivore mechanisms causing convulsions, comas, and even death in herbivores. These effects may not occur in all herbivores. Strychnine, for example, is produced by the fruit of some plants that may be eaten by birds without ill effects, but in mammals, the same fruit causes failure of the central nervous system and induces seizures. The plant reduces herbivory by mammals and the seeds get dispersed by birds that are able to detoxify the strychnine. Grains and seed crops such as wheat and peanuts, which are particularly attractive to animal and insect herbivores, often produce cyanogenic glycosides that release hydrogen cyanide as the tissues are digested. This compound inhibits cellular respiration, thus killing the herbivore. In each case, members of a plant population are consumed by the herbivores, but future generations are spared by the loss. Many phenolic acids act as allelochemicals, that is, the compounds produced by an organism that interfere with the growth or development of another organisms and thus inhibiting root growth of competing species. Many grains are known to release ferulic acid and capic acid into the soil, thus inhibiting the germination of weed species. Phenolics may also act as antifungal compounds, increasing in concentration with fungal infection, thus protecting the plant from further attack. Phenolic compounds produced in tobacco and tomato leaves reduce the growth of these plants' natural predator, that is, tobacco hornworm, without affecting the growth or activity of the hornworm's natural predator. Production of specific combinations of volatiles on the part of the plant signals the predator, which will then reduce further herbivory. The plants have evolved the signal in response to herbivory, and the predators have evolved the ability to detect the signal indicating the location of their host. Terpenoids and aliphatic compounds are often the components of essential oils of plants. The volatile nature of these compounds produces a distinctive color that attracts pollinators. Composition of the volatile compounds often closely matches the natural pheromones produced by the pollinator, mimicking the chemical scent of a female insect in an attempt to attract male pollinators. Pheromone mimicry is found primarily in members of the Orchidaceae, which are often dependent on single species of wasp for pollination. Other plants may mimic the odor of food. The smell of rotting flesh attracted to flies is produced via ammonia and alkyl amines such as cadaverine and putrescine. Methyl esters may attract moth pollinators by mimicking the sweet smell of fruit. Flavonoids often provide color to fruits and flowers and act as visual cues for pollination. Red, blue or yellow in varying patterns stand out against a background of green leaves, helping pollinators locate the flower. Species may have minor chemical differences in their flavonoids that allow for the determination of identity, hybridization between species, and possible coevolution with pollinators. For example, tropical flowers tend to have a more intense red color from anthocyanins than do temperate flowers. This difference correlates with differences in pollinator preference, indicating a role by natural selection. Birds, such as hummingbird, prefer to yellow, whereas bees are not able to discern red but are attracted to yellow. Carotenoids such as xanthophylls and beta-carotene give fruits and flowers distinctive yellow and orange colors. Color patterns are also important in attracting pollinators. Butterflies are attracted to red to yellow color patterns. Flavonoid compounds not only impart color but also may modify color patterns by absorbing ultraviolet light. Now, the types of coevolution. Coevolutionary processes have traditionally been classified into specific coevolution, also called one-to-one, strong or tight coevolution, for example, host-specific parasites, and diffuse coevolution, also called many-to-many, loose, multi-species or guild coevolution, for example, between a guild of plant species and a guild of pollinators. Species by species coevolution occurs in figs and fig wasps. Each of 700 ficus species attracts a unique species of pollinating wasp that raises its brood on fig ovules within the flowering structure and carries pollen to other non-specific fig plants. The mutualism is basically symbiotic. Wasp and fig live in close proximity with each entirely dependent for reproduction on its partner. Such specificity, however, is very unusual. Reciprocal symmetrical coevolution is rare in non-symbiotic flower pollinator or fruit disperser systems where each animal has a choice of foods and each plant is exploited by a range of pollinators or dispersal agents. Coevolution of fruiting plants and dispersal agents where it does occur is often asymmetrical. In contrast to diffuse coevolution, pairwise or specific reciprocal coevolution refers to two interacting species exerting selective pressure on each other, resulting in stepwise coevolution as first one then the other evolves a response to specific change in the other. Pairwise coevolution is probably rare. The strongest potential cases are those in which the host plant species are primarily attacked by specialized insect herbivores. Such a situation may occur in unapparent plants, typically herbivorous species, which have fewer herbivores as a consequence of their small size, relative structural simplicity, and ephemeral nature temporally and spatially. The phylogenetic congruence of the long-horned beetle of genus Tetropus and the milkweed genus Asclepias may be an example of stepwise or reciprocal diversification. By itself, however, comparison of branching patterns of interacting lineages can show parallel cladogenesis, but it does not indicate whether the lineages have evolved. Although diffuse coevolutionary processes are by definition more amorphous, they clearly shape ecological communities because of their ubiquity and immense ecological impacts. In fact, it has been more difficult to find convincing cases of specific coevolution than diffuse coevolution, such as the diffuse interactions between plants and pollinators, predators and prey, plants and rhizobia bacteria, fungi and algae in lichens, and polyps and algae in corals. Dear students, now we will discuss the examples of coevolution. Coevolution can be investigated and understood by studying interactions between two sets of species that represent different taxonomic groups. The mustard family, that is Brassicaceae, is characterized by a group of chemicals known as mustard oils. These are the substances that give the pungent aromas and tastes to such plants as mustard, cabbage, watercress, radish, and horseradish. The same taste we enjoy signal the presence of chemicals that are toxic to many groups of insects. Similarly, plants of the milkweed family, that is Asclepiadaceae, and the related dogbane family, that is Apocynaceae, produce a milky sap that deters herbivores from eating them. In addition, these plants usually contain cardiac glycosides molecules named for their drastic effect on heart function in vertebrates. However, certain groups of herbivores are associated with each family or group of plants protected by a particular kind of secondary compound. These herbivores are able to feed on these plants without harm, often as their exclusive food source. For example, cabbage butterfly caterpillars feed almost exclusively on plants of the mustard and caper families, as well as on a few other small families of plants that also contain mustard oils. Similarly, caterpillars of monarch butterflies and their relatives feed on the plants of the milkweed and dogbane families. The cabbage butterflies evolved the ability to break down mustard oils and thus feed on these plants without harming themselves. Having developed this ability, the butterflies were able to use a new resource without competing with other herbivores for it. Often in groups of insects such as cabbage butterflies, sense organs have developed that are able to detect the secondary compound that their food plants produce. Clearly, the relationship that has formed between cabbage butterflies and the plants of the mustard and caper families is an example of coevolution. Hummingbird pollinators and the red flowered plants that they pollinate represent a classic example of coevolution. Bumblebees, widely distributed species of the genus Bombus, are very important pollinators of both wild plants and important cultivated crops such as alfalfa, clover, beans, and blueberries. Bees, unlike butterflies, have a high metabolic rate and must visit flowers frequently to make an energy profit. To attract these necessary pollinators and to ensure their survival, many species of flowers have evolved the mechanism of either blooming synchronously or growing in landscape patches. Grazing has also been shown to stimulate plant growth and increase net primary productivity. Thus, this interaction has evolved to be beneficial to both the herbivores and the select species on which they graze. Now, we will discuss the importance of coevolution. Coevolution is the process of reciprocal evolutionary change between interacting species driven by natural selection. It is one of the major processes organizing the Earth's biodiversity into interrelated communities of species. The evolution of biological communities is a history of the development of co-evolved relationships. Research in recent decades has indicated that co-evolved interactions form the basis for much of the organization of communities. Primary succession from rock in terrestrial environments often begins through colonization by lichens, which are co-evolved mutualisms between fungi and algae or cyanobacteria. These mutualisms appear to have arisen on many occasions during the history of life, but they are all based upon a relationship in which the fungi provide a habitat for growth of the symbiote and the algae or cyanobacteria provide nutrition through photosynthesis. The colonization of terrestrial communities by vascular plants and some bryophytes also relies upon complex co-evolved relationships including mycorrhizae and rhizobia. The exact role of these relationships in plant and fungal growth appears to vary among taxa, but many of them supplement plant nutrition. Unlike mycorrhizae, which are associated with the majority of vascular plants, rhizobia are genetically complex co-evolved associations between bacteria and legumes or a few taxa and other plant families. These associations are crucial in plant succession in many communities because they allow plants to fix nitrogen into a form they can use for growth. The food webs of communities rely upon co-evolved symbiosis at every trophic level. Plants are eaten by animals which rely for digestion upon co-evolved symbiosis with microorganisms in their gut. Similar symbiosis are required by predators and parasites that feed on other animals. In addition to shaping intimate symbiosis, coevolution also shapes the organization of interactions among free-living taxa. The competition for food or other resources has been shown to lead to coevolutionary specialization by competitors for different resources, thereby decreasing competitive interactions. Predators and prey, flowering plants and pollinators, and birds and fleshy fruits are all involved in complex co-evolving relationships that often link groups rather than simply pairs of species. The processes by which these complex multi-species co-evolved relationships develop are still poorly understood, but there is now little question that reciprocal evolutionary change shapes many of these relationships. Dear students, now we will discuss the criticism of biochemical coevolution. The biochemical coevolution has been criticized on a number of accounts. Some of these include: number one, although insects almost certainly adapt to their host plants, selection on plants by insects is often too weak and variable to drive plant evolution. Second, many plant defenses may be against microorganisms rather than herbivores. Third, conflicting selection pressures from multiple herbivores and plant species may result in diffuse rather than focused coevolution. Number fourth, reciprocal adaptive diversification between insects and plants may derive more from ecology and biogeography than plant chemistry. Number fifth, the predicted increase of specialization through time does not account for the frequent occurrence of derived generalists. The critique of coevolutionary theory between plants and insects became increasingly refined and elaborated over the years. The driving force behind coevolution is the reciprocal selective response of unusually two participants, the plants and insects. Insects exert selective pressure on plants by feeding and forcing them to change, most of all biochemically. The altered plants released from the herbivore pressure, however, will soon be recolonized by the adapted insect species, once again capable of utilizing the new plant species. Then the cycle repeats. The theory opposing the above scenario is that of sequential evolution, which states that plants change biochemically for reasons other than herbivore pressure, such as climatic, soil, or competitive elements. These changes free them from herbivory momentarily. However, the altered plants will be followed by those insects whose genetic variability were fortunate enough to have mutations from which selection would pick up those capable of utilizing the new plant lineages. In other words, whereas insects do not select new plant lineages, plants select for those of insect sequentially. This makes the relationship basically asymmetric. Dear students, this is all about biochemical coevolution. Dear students, we have discussed over here anti-herbivory mechanisms, types, examples and importance of coevolution. We have also discussed the criticism to biochemical coevolution. Thank you very much. God bless you.